Hello, I'm Beatrice Bulan Una with metric number 83362. I'm a crew member of Group 2 and this is my other group members named Waiber Lian Julian, Akila Zahara binti Nur Aizu, Wong Chikai Ifah Akila Ismail and Roland Simon. In this video, I will present about Itchyoplankton activities. First, what is Itchyoplankton? Itchyoplankton are the fish eggs and larvae and mostly found in the upper 200 meters of the water column. Sometimes it is referred to as the near-surface waters. The eggs are inert and only float in the ocean with currents. It is crucial to investigate Itchyoplankton since it has been shown that the quantity of eggs and larvae from different species can serve as a reliable predictor of adult transient spawning population size. It is typically less expensive to estimate the abundance of eggs and larvae in a location than to the sample of the adult. Besides, studying Itchyoplankton is important to monitor fish communities, spawning areas, conservation goals, and population size that spawn. We had a field trip on 8 May 2023 at Pantai Pandan Lundu, Sarawak. There are many activities that we had done related to the aquatic field. One of the activities is Itchyoplankton. In order to proceed with Itchyoplankton activities, it is important to prepare a correct equipment or aquatic instruments that will be used. The equipment and materials that are required to do Itchyoplankton are seed net with 1 mm in size, scoop net, bottle samples, GPAs, pail, and 10% of buffered formalin. For Itchyoplankton activities, there are 3 persons needed to hold the seed net in the water. 1 person will take the coordinate of sampling site and 1 more person will set and record the time. 3 persons will walk backward to pull net in 55 degrees parallel to the beach for 2 minutes. In the last 30 seconds, the person near the shoreline will stop walking and the other 2 persons turning to the shoreline. The coordinates for turning point are recorded. We had done these activities twice. Next, we take out the seed net from the waters and put it inside the pail. We begin to collect the Itchyoplankton using the fish scoop net. It is very difficult for us to see the Itchyoplankton since they are transparent. After collect the Itchyoplankton from the pail and put it inside the bottle sample, we preserve the Itchyoplankton immediately with 10% of buffered formalin. We bring back the Itchyoplankton samples from Pantai Pandan Lundu to invertebrate labs in Unimas. We proceed with the identifications of the family fish larvae. We use red spangle on the fish larvae and observe them under the stereo microscope. We identified that the family of the fish larvae are Megalopidae and Calionidae. We also found that there are 8 in total the number of individuals. That's all of the Itchyoplankton activities that we had done in our field trip. It is very important to use the correct instruments for these activities. Besides, a proper technique is also required to the successful study of Itchyoplankton. So, Itchyoplankton study are important and we had fun when learn about the Itchyoplankton. That's all from me. Thank you.